This video is going to talk about active transport. So this is basically going to be the opposite of passive transport. Sometimes we want to move materials against the way that they want to go, so against that concentration gradient. And so active transport will be moving materials against a concentration gradient. So instead of going high to low concentration, we are now going, low to high. And because we're moving something against the way that it wants to go, it will require energy. This is like if we have a hill and we have a shopping cart. Okay, if I have the shopping cart at the bottom of the hill going high to low, and I let it go, it will take literally no energy on my part for the shopping cart to go from high to low, the direction it wants to go. If, on the other hand, I have my shopping cart at the bottom of the hill, and I want to move it from low to high, it will take a significant amount of energy for me to push it from low to high, a direction opposite of where it wants to go, and that would be active transport. It required energy to go from low to high. So there's a couple different types of active transport. One of those is when dealing with small molecules. When dealing with small molecules in active transport, we will use membrane proteins, but not channels this time. We're going to use protein pumps. If you think of that word, and if you see that word on a worksheet or a test or a quiz, that word pump should imply to you something using energy. So cells will use protein pumps to concentrate certain substances in specific places. That is a reason for doing active transport. You might have been wondering, why move something to a high concentration if it doesn't want to be there? Well, we might have a purpose for putting a whole bunch of things in a specific place, because we might use that for something. And so here in our picture we have low concentration and moving these red molecules to an area of high concentration. And notice what it's using here, and we haven't talked about this yet, but what it's using is ATP. And ATP is basically cellular energy. And so you'll know that active transport has occurred if ATP is used and turns into ADP and PI, which is outside of the realm of this video. Another type of active transport is bulk transport. In bulk transport, just like what it sounds, really large molecules or solid clumps of material will be transported. And there's basically two types of bulk transport: into the cell and out of the cell. Endocytosis, think about what we hear there, endo meaning in or inside, cyto referring to the cell. In endocytosis, we take material into the cell. And so in endocytosis, we form a vesicle or vacuole within the cytoplasm that brings in this item. So these are sort of images of active transport in terms of endocytosis. The food particle comes in, we sort of create like a Pac-Man-esque thing here. The cell closes around the food particle, ultimately comes together and pinches off, and you then have the cell membrane back intact, and then a food vacuole or vesicle, which might then merge with a lysosome to break that food particle down. Real life example of endocytosis, of course, a white blood cell. Attempting to basically eat some sort of invader cell. Okay, this is just a nice picture of multiple molecules and our phospholipid bilayer. And so notice that the vesicle itself then has a phospholipid bilayer as well at the end. The opposite of endocytosis is exocytosis, that same cyto referring to the cell, and exo referring to exit or something outwards. It's like existential or extraterrestrial. And so in exocytosis, the membrane of a vacuole fuses with the cell membrane, forcing the contents out of the cell. It is literally endocytosis in reverse. We start with the vesicle, it fuses with the cell membrane, and then pushes its contents into the extracellular space, the space outside of the cell. And this is, of course, a really big deal when talking about neurotransmitters and signaling between neurons. We don't just like one or two sort of signals, like maybe you should move your arm or something like that. Maybe you shouldn't. We want a whole big signal and we want it to happen fast. We don't want to wait for single things to go through individual channels. So we will use exocytosis to bulk transport neurotransmitters out of one cell towards another.